Hey there, everyone! How's it going? I hope you're all having a wonderful day. I'm super excited to have you back with me, Dr. Petsavan, here at the Shed channel. Today, we're diving into a fantastic online lecture all about crafting the perfect methodology section for your manuscript, ideal for those looking to get published in international journals. If you're not already part of our awesome community, make sure to hit that subscribe button so you don't miss out on any of our exciting updates from the Shed Luminous. All right, let's jump right in and get started. So, let's break it down. The methodology section is like the recipe for a successful study. It tells us how the research was conducted, and why that matters is because it allows us to evaluate how valid and reliable the findings are. Think of it as giving us the power to see if we could recreate those results ourselves, just like when you try baking a cake from a recipe. If the instructions are unclear or missing, you might end up with a cake that's more like a pancake, definitely not the outcome we're going for. Now, if you've ever read a research article, you know that the methods section is where the magic happens. It's where you lay out how you conducted your study, the who, what, why, and how. Think of it as your roadmap for readers, guiding them through your research process. A well-crafted methods section not only showcases the rigor of your work, but also allows other researchers to replicate your study, which is vital for scientific progress. The key thing to remember here is that researchers need to provide enough detail for others to replicate their work. It's all about balance, being thorough while keeping things clear and concise. We want to know what was done without wading through pages of jargon, right? Typically, you'll find this critical section right after the introduction of a manuscript and before the results. It's a vital piece of the puzzle for peer reviewers and fellow researchers who are trying to make sense of the findings. So, next time you read a research paper, take a good look at the methodology section. Appreciate the effort that goes into ensuring that studies can be trusted and replicated. It's an important step in the scientific process, and it helps push knowledge forward. Let's talk about the structure of our methodology section. Think of it as the heart of your research project. It needs to be organized and clear so that everyone can understand exactly what you've done. We'll be using subheadings to guide us through key components, including study design, population, sample size, data collection methods, and a whole lot more. It may sound like a lot, but don't worry, I'll break it down for you. Starting with study design, it's super important to specify what type of study you're conducting, whether it's a randomized controlled trial, a cohort study, or perhaps a cross-sectional one. And remember, you have to justify your choice. For example, if you're looking into the incidence of diarrhea in children under 5, a prospective cohort study can provide the timeline and data you need. Next up, we have study population and setting. Here, you'll want to clearly describe your participants. What are the criteria for including or excluding them? And don't forget to share where you're conducting this study. For instance, if you're gathering data from adults with type 2 diabetes across clinics in Luang Prabang, that gives a nice geographic context. Another example is, data were collected in 3 provinces of Lao PDR, representing 3 regions of the country, with a high number of populations. VNT Capital, VTE, the capital city in the central region, bordered with Thailand with a population of 836,000 2023. Then, we move on to sample size and sampling. Calculating an appropriate sample size can feel daunting, but it's crucial for ensuring your findings are valid. Make sure to explain how you selected your participants and which sampling methods you used. This can really enhance the credibility of your research. For example, a sample size of 200 was calculated to detect a 10% difference in blood pressure with 80% power and alpha equals 0.05. Another example, a multi-stage sampling was used. First, the districts were purposefully selected. Second, villages were selected using probability proportional to size sampling. Third, the households with children under 5 were selected according to the list of households given by Chief of Village. Let's move to data collection methods. Here, you'll want to detail the tools and procedures you used. This could be anything from questionnaires and interviews to lab tests. Make sure to mention the reliability and validity of these instruments. For example, you might say, the SF36 questionnaire, validated in reference, was used. It adds credibility. Next, you'll describe your data collection process. Every step you took, including timelines and protocols. Did you provide training for your data collectors? What quality control measures did you implement?
These details enhance transparency and trust in your work. Remember, clarity is key. Moving on, let's tackle variables and measurements. Here, you need to define your primary and secondary outcomes clearly. Explain how you measured these variables. For example, you might say, hypertension was defined as systolic blood pressure greater than or equal to 140 mmHg, or diastolic greater than or equal to 90, measured using a calibrated Omron N6 monitor. This specificity will help readers understand exactly what you studied and how you assessed it. Now, if you're working on experimental studies, you'll want to describe your intervention. What did the participants receive? For instance, participants received 500 milligrams metformin daily for 12 weeks, while the control group received a placebo. If it's an observational study, explain the exposure and its measurement. For those dealing with lab work, don't forget the sample collection and storage. This is a crucial part because it ensures our data is reliable. When you're describing how samples like blood, tissue, or swabs are collected, think about the tools and conditions you'd use. For example, you might say, I collected nasopharyngeal swabs using sterile flocked swabs and promptly stored them at -80°C in viral transport medium. This way, you're being specific. You're telling readers exactly what tools you used and how you maintain the integrity of those samples. Plus, it helps others replicate your work. Now, let's move on to storage conditions. Make sure to specify things like temperature, duration, and any preservatives used. This ensures that anyone who reads your manuscript knows how to properly handle samples. After collecting our samples, we need to process them before analysis. This can involve various steps like centrifugation, dilution, or DNA extraction. To illustrate, you might say, blood samples were centrifuged at 3000 RPM for 10 minutes at 4°C to isolate plasma. This gives your audience all the necessary details about what you did and the conditions you followed. Don't forget to include any reagents or equipment you're using along the way. If you mention specific pH levels or temperatures, that adds more scientific credibility to your writing. Laboratory or microbiological techniques. Here, you'll outline the methods used for your experiments. Whether it's PCR, ELISA, or bacterial culture, be sure to be clear. For example, you can say, bacterial isolates were cultured on Mueller-Hinton agar at 37°C for 24 hours. See how that provides insight into the method used? It also allows others to follow your protocol accurately. If there are specific incubation times, temperatures, or staining methods involved, make sure to highlight those. Detail is key here, my friends. Let's talk about the equipment and reagents section. This is where you'll list out all the specific tools and materials you used in your experiments. It's like setting the stage for the great show that is your research. When you mention equipment, be specific. Include the brand and model. For example, you might say, real-time PCR was performed using the Biorad CFX90 system with TACMAN probes from Thermo Fisher. CAT number, insert your document number here. This clarity helps others replicate your work, which is super important in science. Now, let's head over to quality control and validation. This is where you establish the reliability of your methods. You want your readers to know that you've taken every possible step to ensure accuracy. Talk about the controls you used, like positive and negative controls and calibration standards. For instance, you could say something like, we validated assay sensitivity using known concentrations of pathogen. This kind of detail really strengthens your manuscript and shows that you care about the integrity of your research. Next up, we have data recording. This is where you outline how you measured or recorded your results. It's all about making sure that your findings can be understood and utilized by others. For example, you might share, colony-forming units, or CFU, were counted manually after 48 hours of incubation. This not only informs your readers about your methodology, but also gives them a peek into your process. All right, first up, data analysis. This is where you explain how you managed your data, and it's a key part of your manuscript. Let's start with the basics. How did you manage your data? You'll want to outline the entire process, from entry to cleaning and storage. For example, you could say something like, "The data were entered using Excel." Try to be specific about what tools you used because this helps readers understand your methods. It's like giving them a behind-the-scenes peek. Then, highlight the statistical or qualitative analysis methods you applied. For instance, we analyzed the data using Stata software version 12. Make sure to mention any comparisons or analyses you did, like t-tests or logistic regression. This is crucial as it shows how you determined your findings. Don't forget about handling missing data. Readers are going to want to know what you did in those situations. It adds credibility to your work. Here's a quick example.
The data were entered using Excel and analyzed using Stata software version 12. Continuous variables were compared using t-tests, and logistic regression adjusted for confounders was performed to determine the associated factors. Or, if you're into qualitative research, you can say something like, "The analysis took both thematic, deductive, and inductive approaches that allowed us to delve into preconceived themes, deductive, along with data-driven themes, inductive." Now, let's talk about ethical considerations. This part is so crucial and shows your readers that you take the moral responsibilities of research seriously. First, make sure to mention your ethical approval, if you had to get one. For instance, you might say, "The study was approved by the Research Ethics Committee of the University of Health Sciences with approval number 618/REC on August 30, 2023. It adds a layer of professionalism. Then, talk about informed consent and confidentiality. It's super important that you outline how participants' privacy was protected. You can phrase it like this: "Prior to participation, written informed consent was obtained from all participants. For adolescents under 18 years old, written parental/guardian informed consent was obtained for their participation." This shows you respect your participants' rights, which is absolutely vital in research. Before we wrap up, here are a few best practices to keep in mind. Be clear and precise. Use straightforward language. Avoid jargon unless it's absolutely necessary. Use past tense. Describe what you did, not what you do. Phrases like "participants were recruited" sound much better than "are recruited." Now, every journal has its own guidelines. Seriously, check those author instructions for things like word count and structure. Following the target journal's guidelines is a must. Think of it as your roadmap. Once you have that down, organizing your section is key. Use subheadings to break up the text. It could be something like study design, data collection, and so on. This makes your methodology easier to navigate, super important for readability. Also, adhere to reporting standards. If you're doing a randomized controlled trial, look into the consort guidelines. For observational studies, check out the Strobe guidelines. These are like your best friends when it comes to ensuring you're covering all the necessary details. Now, here's a common pitfall to avoid. Don't mix in your results or discussion in this section. Stick to the procedures and protocols only. Keep it straightforward. Make sure you provide enough detail for anyone to replicate your study, but don't drown them in unnecessary minutiae. Let me share a few common pitfalls to avoid. 1. Lack of detail. Be specific. If you used a questionnaire, mention the version. 2. Irrelevant information. Focus on methods that are actually relevant to your research question. 3. Ambiguity. Clearly define your terms. For instance, if you say moderate exercise, it should be quantifiable. 4. Missing ethical details. Always include information about IRB approval and consent processes. Ethics is super important. 5. Inconsistent terminology. Stick to one term throughout. Are they subjects or participants? Pick one and go with it. For those of you working in a lab, there are some additional tips. Always provide detail on your methods. Avoid vague terms like samples were processed. Instead, specify the methods and conditions used. And never forget to describe your controls and quality measures. This helps anyone trying to replicate your work. Now, let's talk practical tips. If you're a student or just starting, here's what I suggest. Start with an outline. Draft subheadings and bullet points before diving into full sentences. Reference similar studies. This can give you a clue on how to structure your methodology. Don't be shy. Share your drafts with peers or mentors for feedback. Use reporting checklists like Consort or Strobe. They're super handy for ensuring you've covered everything. And here's a fun exercise. Try rewriting a published studies methodology in your own words. It's great practice. Here's a quick sample of a methodology section for context. Study design, randomized controlled trial conducted from March 2023 through June 2024. Population, healthcare workers aged 25 to 55 with moderate to high stress were recruited into the study. Intervention, participants were assigned randomly to an 8-week mindfulness-based stress reduction program or a placebo group. Data collection. We used the Perceived Stress Scale, PSS-10, assessed a baseline and after 8 weeks. Analysis.
We used repeated measures ANOVA to examine the differences pre- and post-intervention using SPSS V27. Pretty neat, right? Here is the summary table that can guide you easily. Let's have a short activity. Take 10 minutes to write down a methodology section for a given study. For example, cross-sectional study on hypertension in rural adults. For lab, the E. coli example, drafting a laboratory process section. Here are the resources you can read further. Enjoy reading. To wrap things up, remember, your methodology section is the backbone of your manuscript. Be clear, detailed, and make sure your writing is reproducible. Always follow journal guidelines and reporting standards. Practice makes perfect, so keep seeking feedback. So there you have it, a comprehensive guide to writing the methods section of your manuscript. Remember, the methods section is your chance to showcase your hard work and contribute to the broader scientific community. Keep it clear, keep it detailed, and don't shy away from sharing your unique journey. I hope this gives you a clearer picture of the methodology section. Thanks for joining me today, and I can't wait to see the amazing research projects you all create. Let's keep the conversation going. Feel free to drop your questions in the chat, and I'll be here to help. Thanks for listening, and I can't wait to see you all at our next discussion. Don't forget to like, subscribe, and share your thoughts below. Let's keep learning together.